Now let's look at some types of chemical reactions. We have some overlapping terms here, different terms that can be applied to the same reaction. So you already learned about hydrolysis and dehydration synthesis. Remember, that was with the you are what you could eat lesson. So you also know about polymers and monomers. In the top picture here, we are starting with a polymer. Notice it's two individual monomers linked together. We're going to break that bond and end up with separate monomers. So if we use a term you already know, this would be an example of a hydrolysis does this. Hydrolysis is a type of decomposition. So if you think of decomposition, it just means something breaks down. Decomposition is any time that you are breaking the bonds. So hydrolysis is a type of decomposition. It's decomposition in which you use water. Then we have the word catabolism. Metabolism is the chemical reactions of your body. And metabolism can be split into two types, catabolism and anabolism. So when we use the word catabolism, it again means breaking down. It's a type of decomposition, and in context of the body. Like we talk a lot about protein catabolism, means you're breaking down proteins to get amino acids. Then we have exergonic. This has to do with energy. For the EX, think of exit. Exergonic, energy exits, or you release energy. We just talked about ATP and how when you break that high energy bond that holds the third phosphate on, it releases energy. That's an example of exergonic. So whenever you break bonds, you release energy. Then we also have exothermic. Think of therm. Therm means temperature. Remember the second law of thermodynamics we just talked about? When you use ATP, you get heat. Whenever you break bonds, you release heat. So you can tell that you have a decomposition reaction going on because temperature will increase. If one of these reactions is happening, the temperature will go up. It'll get hotter. Then the other side of this, we have synthesis reactions. So you already learned in the You Are What You Eat lesson about dehydration synthesis. Anabolism, remember, is the other type of metabolism. So this refers to it happening in the body. And here we have our monomers that we're starting with, and we are building a bond. So we end up with a polymer, and there is the new bond that we just made. And whenever you do this, when you're building bonds, that is endergonic. So think of the EN for enter. Energy enters. So you have to use energy to build bonds. And whenever you're using energy, that is endothermic. So now this time it's temperature enters. So you use up energy, you use heat. And because you're using it, heat is like money. Once you use it, you don't have it. So temperature goes down in these types of reactions. Another important thing to go along with reactions is enzymes. Enzymes are catalysts. This means they speed up reactions. So you have metabolism, chemical reactions in your body. You have catabolism, you have anabolism. These are constantly happening. They keep you alive. Enzymes speed them up. So you have all these reactions going on in your body. Remember, that is metabolism. Metabolism just means all the chemical reactions in your body. You need them to happen fast enough to keep you alive. Chemical reactions will eventually happen on their own, but it would be so slow that you would die waiting for them. So you need them to go fast. So you have enzymes in your body. The enzymes make the reactions happen fast enough to keep you alive. So enzymes are proteins that your body makes. You make your own enzymes to speed up your reactions. And you rely on your enzymes to stay alive. So we're going to look at enzymes and how they work. And it's important to keep in mind that they are proteins. Remember, proteins function based on shape. And remember, proteins can denature, which means change their shape and lose their function. If they get too hot, or if you change the pH. Right? Think about like cooking food. We talked before about a cooked egg versus a raw egg. Raw egg is clear and runny. You cook it, it's white and solid. You denatured the proteins in it. You changed their shape. So we look at an enzyme now. Here's our first example. Here's the lactase enzyme. It is used to break down lactose, which is the sugar in milk. So here we have a polymer. Right? It's the two molecules linked together. Notice the shape. They have a certain shape. The enzyme here has a shape that matches. This is the active site. So an enzyme has an active site. This is where the molecule fits. And enzymes are specific. Like how puzzle pieces fit together. Think about when you're doing a puzzle, you can't just put any two pieces together. Only the two that fit together can go together. It's the same with enzymes. Only lactose can bind to the lactase enzyme. Their shapes have to fit to each other. This is why there are people who are lactose intolerant. Some people don't have the lactase enzyme. And if you don't have that one enzyme, you can't digest lactose. You can't just use any old enzyme to digest lactose. Only lactase will digest lactose. So enzymes are specific to which molecule they will bind to. The molecule that it's going to bind to is called a substrate. So you have an enzyme and it fits to a substrate. So they bind to each other. So here you can see in this picture, the molecule is bound to the enzyme. This is called the enzyme substrate complex. Once they are bound to each other, the next thing that happens is the enzyme bends. Look at the shape of the enzyme. See it here compared to here? It bends. And this is not denaturing. It's bending on purpose. That's how it works. When it bends, it breaks that bond. So this bending is called induced fit. So it bends and breaks the bond. Now we have two individual monomers. Whatever is released from the enzyme is called product. And these are released. So this is now free to go into your blood from your digestive system. The enzyme goes back to its original shape, so it unbends. And then notice we have an arrow here that it goes back to the start. It's ready to go again. So you can reuse your enzyme. Let's look at another example. Here's our enzyme. And notice here, these are the active sites. Here is our substrate. Substrate is the molecule that fits in those active sites, and this is glucose. These are glucose molecules. Then here we have the enzyme substrate complex, so the glucose molecules are in the active sites there. They fit together like puzzle pieces. Here we have the induced fit, the enzyme bends. And this time when it bends, it forms a bond. And then we release the product. And then the enzyme unbends and it's ready to start again. So the basics here, you start with a substrate. Substrate is what you start with. It binds to the enzyme. The enzyme bends, it either breaks the substrate or links two substrates together, and you get product. Product is what you end with. And then the enzyme is ready to repeat. So you can reuse the enzymes, but there is a limit. They don't last forever. Like anything else, they wear out, and then your body needs to make new ones. So even though you do reuse enzymes, you also have to constantly make new ones. So they do wear out, and you make new. So every day your body is making new enzymes. Another thing to think about with enzymes is they work by bending. That bending is key. It's when they bend that they form or break a bond. Temperature affects ability to bend. If something is cold, it is stiff, and it will resist bending. It's hard to bend something that is cold and frozen. If you warm it up, that makes it more flexible, and it will be easier to bend. This information is going to help you with your lab. Remember this, the effects of temperature on the ability of an enzyme to bend. And if we tie this in to what we had up here with types of reactions, this top one here, you're breaking bonds, so this is a decomposition. And the bottom one here, we're building bonds, so this is an example of synthesis. So enzymes work in both decomposition and synthesis reactions.